All right, should we get started?
Do it.
Wonderful. Well, we're super excited. We know a number of people are here. We're very excited that you have come today to our fireside chat. And one of the things that we're hoping to do, we know that we rented an Airbnb. It's really amazing. There's 4 separate rooms that all have the same thing. And we're very excited to share this with you. So we know it's been a year for many of you doing webinars. And so we want to keep this one to be kind of fun. And what's more fun than hearing all of the war stories and in the trenches and maybe some of the horror stories of dealing with wearable data. So our motto for today is to be much more show, don't tell. You have 3 of the people that I admire the most in the industry who have been in the trenches working on these different types of tools. So what we're going to do today is each of us is going to go through, we're going to introduce ourselves, and then we're going to ask you to introduce yourselves. So the 4 of us will do an introduction, then we'll have a poll for you to introduce the roles that you're in. After that, we're going to go into some themes around the playbook, which is an open access guide that you'll be able to use. And then we'll be sharing different stories about wearable data gone awry. So I'm Andy. I'm the CEO and co-founder of Elektra Labs, and I'll be the moderator for today's session. Kate.
Thanks, Andy. Good morning, afternoon, everyone. My name is Kate Leiden. I am the Chief Science Officer at VivoSense, which, if you're not familiar with VivoSense, we are a data analytics and consulting company. We specialize in data from wearable sensors, physiological monitoring, behavioral monitoring, and mostly in regulated clinical trials. Before joining VivoSense, I spent a lot of time in the academic research setting. So I have quite a few war stories of data gone awry. So excited to chat with everyone today.
And then what we'll do here is we'll have you do popcorn style. So you get to pick the next person.
Yeah. Jeremy, you're up.
Thanks, Kate. Hello, everybody. Thank you for taking some time to spend a few minutes with us today. We really appreciate you dialing in. My name is Jeremy Wyatt. I'm the CEO of Actigraph, and very excited to be here. Actigraph, our mission is to bring life to digital data. And so we're excited to talk to you about where we've not done that today and talk about some of the war stories that we've learned along the way. But again, thanks everybody for hanging out with us today. Shamal, go ahead, buddy.
Everyone, my name is Shamal Patel. I am an engineer by training. I've been working in the space of wearable technology for about 15 years, most recently at Elektra Labs. And before Elektra Labs, I was at Pfizer leading the data science team, where we brought multiple digital endpoints from an idea to deployment in a clinical trial. So I think over these 15 years, I've been in the trenches quite a bit, and I think I've seen a fair bit of things, although I do find new and interesting ways of getting in trouble with data. So that's always an ongoing topic, and excited to be here with the panelists and to talk about some of these stories. So
great. So now we're going to ask you to do your intro. So there'll be a poll that comes up. You'll get your first set of polls. And we'll give you about 20 seconds to fill this out. If you're unsure, also pandemic is having you like really rethink parts of your career, you can always pick other and talk to us like separately from this session. Everybody
on here is hiring, right, Andy?
We're all hiring. We're all hiring. Come join. There's way too much work. Look at our job boards. Great. And then we're really thankful for the team that's been helping us put together this session. So they'll let us know when we kind of have enough answers here. Or does that show live? I'm not totally sure.
Yeah, we'll just transition over whenever you're ready.
Okay. All right, let's see the results.
Wow.
Okay, great. So lots of people here from academic institutions, which is super exciting, and we're really excited to. There's really a broad range here, both for people who are doing this sort of work in clinical research, but also I think many people are starting to do this work more in clinical care, and so we're excited to have you here. We have a second poll now that would like for you to do. All right. So far, we saw there's some, as you're answering this, just to kind of create more of a dynamic where we can't have everybody in this very fancy Airbnb that we're all in, but we would like to have some sort of interactions more with the team. So there's two ways to do this. One is there's a chat. We'll loosely be looking at the chat, but if you really have a specific question, please start asking it in the Q&A. We're going to go through and answer questions that we have, but then we're going to pull in questions from the Q&A. All of us, I believe, are on Twitter, and somebody stop me if there's a question that if you're watching this on a recording or you want to communicate with us. I think some DMs are open and you'll be able to also communicate there or send us notes in other formats. So can we please open the professional role? Okay, lots in research. Wonderful. All right, so what I'm going to do now is I'm going to help. There's so many different war stories, and there's so many different ways people use wearables and remote monitoring that I'm going to put some framing, and then we're going to use this framing as a path forward here. So what I'm going to do is, this is an open access resource. I'm going to share my screen. And for people who don't know, the Digital Medicine Society is a 501c3 nonprofit. If you're not part of this society, I highly recommend you join. It is for a professional society for those who practice in the area of digital medicine. That could be for researchers, for engineers, for security researchers, for clinicians. It's a broad range. And last year, a number of people came together to say, okay, how are people collecting measures digitally across research, care, and public health? This is a 400-page resource. I will definitely not be going through this with you today, but what we will be doing is thinking about things about what are these different types of technologies and where do they play? So there are really three things that matter a lot. So really thinking through, what do you want to measure? Then once you know what you want to measure, what tech is right for that measurement? How many people have been in a meeting where someone's like, I need to use an Apple Watch, and you're like, but to do what? And then what are the different operations that are important for doing deployments? And then one last framing that I'd like to note for people is there's a number of different things when we talk about technology. So there's the underlying sensors, then there's these algorithms that are deriving information from the sensors, and then that whole data system needs to get put into the data platform. We will not be talking about this third aspect today. We will be talking a lot around sensors and the data platform. If anyone hears this, my two cats are fighting. And so I apologize if that ends up being very loud. So now that we've thought a little bit about this, Kate, do you want to start thinking through and helping the team think through some of the questions that you start having as you're thinking about measures?
Sure. So I think when we think about measures in, specifically digital clinical measures, there are certain things that are exclusive to digital, but when we're selecting a measure, a lot of the same principles apply that if we weren't using digital. So selecting the right measure is obviously at the utmost importance. As an example, we often think about measuring activity, and people's default is to select moderate to vigorous physical activity, MVPA, right? Everybody's heard of MVPA. That's kind of the default outcome measure that somebody might select. But if we're interested in monitoring a clinical population, for example, a population with COPD, at baseline, patients with COPD probably don't spend much time in moderate to vigorous physical activity. And so if we've selected change in MVPA as our outcome measure, the chances that this treatment benefit they'll increase their activity in moderate to vigorous physical activity is low. So they may increase their time in light intensity activity, But even healthy populations don't spend much time in moderate to vigorous physical activity. So, just from a starting point, we need to be really thoughtful about, is our outcome measure relevant for this clinical group? And so that's just a rough example of kind of some of the errors that I see happening in digital, where people don't understand all of the measures that are possible and how those measures relate to their clinical population. So I think just being really thoughtful about that from the beginning is really important. And I think that Jeremy probably has some thoughts around MVPA as well.
Yeah. I think what we often see is people will jump into the MVPA conversation, which I think you said the acronym, Kate, but it's moderate to vigorous physical activity. It's this notion of bend activity level. So you just think about your day, and anybody has an iPhone, on Sundays it pops up and says how much time you spent on your phone. So MVPA is sort of the same way. It's a series of these categories of time in which you spent you spent time in these different light activity timeframes, or moderate or vigorous. And so oftentimes we'll have folks come and they'll say, hey, we want to use that endpoint. We read about it all the time in literature. And for all the researchers who are on today, thank you for your excellent work that you've done and helping us develop those. But gosh, I usually say to them, well, which one do you want to use? And they say, well, I want to use the MVPA one. And that's where it starts. The trouble starts, because people miss the fact that those are oftentimes, they vary. The measure varies depending on exactly what we're trying to do in the study. And as part of the issue, what we're talking about today is that there's a general concept of using wearables in clinical trials that really causes us to have a disconnect sometimes with what the sponsors expect and what we're going to be measuring. So anyway.
Yeah, and
but Jeremy, I was going to say.
Go ahead, Andy.
Yeah.
Jeremy, step's a step, right? Like they're not really all that different across the way people walk.
Yeah, I hate steps, but no, everybody's steps is different. The best example I give of that is, I've got a 10-year-old or 9-year-old, and his step does not look like my grandmother's step, right? I mean, that's the two ends of the spectrum. And so, how we qualify and quantify that it makes a difference in the measure. And we've seen folks do that at some of our horror stories, where we've seen our groups come in and just take that carte blanche step method and really get upset because they were disappointed that the steps don't seem to correlate with the activity levels. And so when you think about activity monitoring, we're talking about an accelerometer on a device. And in that case, the example there is, we measured these steps using a fixed algorithm where we turned raw accelerometry data into an outcome measure that was steps, and it didn't work out right. Fortunately, in our case, we were able to kind of backpedal because we had collected the unadulterated, raw data. We could actually go back and reprocess that. So it's a good example of a war story we've seen with steps, Andy.
I have something to add there. A story of my own. So one of the things that we've worked on was to kind of build a foundation for measuring gait across multiple patient populations. And so this is an open source package that we released. And what happens is that when you develop certain capabilities, you develop familiarity with, working with specific technologies, then it almost becomes like a hammer looking for a nail. So you know how to do something really well, and you have prior experience. And so when you look at a new population, you often start looking at it from the same lens and saying, okay, can we apply this, and can we just go with this, right? So it becomes sort of more of a default. And the example is essentially a lot of the work that we did on Gait was motivated by neurological patients who have neurological disorders like Parkinson's. And and thinking about sort of the population level differences, Gait in Parkinson's is very different than Gait in somebody with stroke or ataxia or some other condition. Just the manifestation, the type of impairments, those are different. So there are similarities, and you can kind of leverage those. But if you take a tool that has been validated in patients with Parkinson's and just assume that when you apply that in patients with cancer, for example, to measure impact on their gait, like gait speed and things like that, things might not translate The tendency is very much to kind of use what we know and have learned and have had success with. But what we often found is that, by trying to essentially replicate the same technical setups across different conditions, we were discounting on these differences and not really fully understanding, what is it that is truly important for a specific condition.
Yeah, and so before we move too far away from the conversation about steps, I'll just share my screen real quick to show a visual of what Shaimel is talking about. Can you guys see that?
Yep, we got it.
Yeah, so this idea that we're talking about analytical validation here, and this idea that Shaimel kind of articulated where we have a solution and we try to find a question to fit that solution to is kind of a backwards way to approach this. We need to come at it from what's our question and then how do we find the appropriate solution. And so what I'm showing on the screen here is an image of normal healthy gait at the top, what the fall pattern looks for normal healthy people, and then several clinical gait populations and what that footfall pattern looks like. And so you can imagine that if we develop and validate an algorithm on a normal population, a healthy population, and then we try to take that algorithm and apply it to a clinical population, the idea that we'd be able to accurately detect steps becomes significantly reduced. We need to evaluate, what are the differences between the different populations and how those patterns and stepping might impact the accuracy and precision of our algorithm. And so it's just, it's not a one size fits all approach. We can't take our solution that we applied over here and apply it over here and expect that we're going to have the same results. So we need to be really thoughtful about, population specific analytical validation techniques and approaches.
Hey, I've kind of been hearing more and more people talk about this. Can you tell us a little bit more when someone says, my favorite question, is this validated? How do you think about validation? What are the different parts? Where does analytical validation fit in?
Yeah, so that is obviously a loaded question, right? Like, is this validated? And I think there's so many different parameters around that you need to also ask, is this validated in this population, in this specific setting to, measure X? Like it needs to be a very, very specific question. And then equally, when you're evaluating, validity evidence and deciding, is this existing validation evidence sufficient for me to use in my population? You need to think of all those questions again. So it's kind of, if I take this method that was developed in this very narrowly defined use case and apply it to my use case, what are the chances that it will, what are the potential pitfalls, right? Like what's different about my use case that could potentially impact the accuracy of this algorithm or this method, this approach?
Yeah, I think that the second aspect of what you touched upon, Kate, the settings in which the methods have been validated, that's another important aspect to consider. This is pretty common, and just looking at the poll in terms of the makeup of people's backgrounds, a lot of people are from sort of the academic background, and a lot of the validation work, early sort of analytical clinical validation work, happens in academia, typically happens in sort of a lab type of setting, and which makes a lot of sense because we can control things in the lab, we can do things very fast. We also have really high quality ground truth. And so the process of bringing a new digital measure from being validated in a lab, sort of controlled setting to the home, there's so many kind of things that can go wrong in that process, right? And I think as researchers, we have tried to account for these by introducing more flexibility in our protocols, kind of thinking about what types of situations we might encounter at home, and kind of bringing those in. But still, it's just impossible to account for all those things that happen in real life. So one example I have is in some work that we did where we were looking at sleep, we had, like everybody does, we validated the methods in a sleep lab using PSG as the ground truth. And then we, deployed the devices at home. And when we started getting the data, we started seeing some odd sort of behaviors. People getting up at night, going to the kitchen, maybe they're to drink water, or going to the bathroom or whatever. But things that we weren't seeing in the lab, because lab is a sleep lab is a constrained setting. People are wired up with electrodes and stuff, so they might actually not have the incentive to move around, even if they wanted to. And so as soon as we started getting that data, we had to sort of figure out, okay, how do we account for this? And then from an analytics point of view, we had to sort of think about how do we structure analysis to identify these periods and break the sleep periods into chunks that can be analyzed as sleep and discount for these sort of awakenings that are happening. And sometimes this is like, until you get the data from a deployment, it's just hard to anticipate all of these things. And in some cases, there's not much you can do, especially if there are behaviors that that you don't have any explanation for or a way to deal with. And I guess, sort of through this iterative experience, you can improve and refine, but I think whenever we have deployed devices in the real world setting, there has always been surprises.
Oh yeah.
Yeah. So one thing we're going to continue doing here, and you'll see, is we're going to give you some theory, and then we're going to shift back into war stories as we're going through. So some of the things that we just talked about, there's a paper called the V3 paper, which is verification, analytical validation, clinical validation. I just dropped it in the chat. It's one way to think about these. Shamal also talked about one of the pieces that he published. I think, Shamal, that was on GitHub that people can share, so we can send that out. The thing that I love about what all of us are working on is generally people don't compete that much on endpoints and on measures, right? We don't have Pfizer blood pressure, Novartis blood pressure, AstraZeneca blood pressure. You just have blood pressure, right? So as we start thinking about steps and activity and sleep, like there's a lot of collaboration here where people are all here to learn from one another. Sometimes as we start introducing some of these war stories to you, sometimes it might be the tech related. Sometimes it might be the context that they're in. Sometimes it actually just might be how you set up the tech, right? Jeremy, you think a lot about like, what are some of these setups? How are you thinking through, like, what are some examples of things that people should think about, especially as they're starting to deploy these sorts of tools and want to get high quality data in their systems?
Yeah, it's a great question. So, on the operations side, I mean, I think we tend to generalize the idea of measuring, with the digital sensor. And I saw a question in the chat about, how do we handle, using multiple digital sensors for a single study? I haven't figured it out yet, but I think that some examples that point to the need to think through it wisely before you ever start. We had a study that they literally wrote in the protocol, they required the patient to wear the device 8 days before a visit. Now, I'm not sure, what the justification was there. I think they wanted, they knew they were coming into a lab and they wanted to get the data from them at that time. But what they had not thought about were the logistics around doing that, right? Like, what if the device has been sitting on the shelf for a month before that? Who charges it? How do you do that? So we addressed that by, helping them set up reminders to call the patients to do that. But we also basically said, look, if you didn't get it, 8 days prior, we can continue to capture it 8 days after. And it's got to be convenient. It's just got to be easy. And the setup of the device and the product and setup of the sensor, that's key. And being able to sort of change that, midstream. In a lot of cases, we find when the consumer technologies are used, they do well, but the actual setup of the product, tends to vary over time, especially if you've got firmware differences. And Lord, I've seen those stories happen. I've got examples of that, but I don't want to hog all the time. Is that what you were thinking, Andy? Is just around how do we kind of do that? And
yeah, maybe for people who don't know, do you want to tell them a little bit about like what firmware is and why that matters and how that might? I'm sure that Kate and Shamal also have some firmware stories too.
Yeah. Yeah, so the firmware is the software that runs on the actual device itself. So if you're wearing a Fitbit or an Apple Watch or even one of our products, it has its own set of software that was developed by some engineers that essentially is designed to show things on the watch like at the time, but also for our devices, it's designed to accurately capture and process data, from the accelerometer that's on the device itself. And so, in some cases, in the past, before we figured out how to do this the right way, we had different preferences around what that firmware could do. So we had a lot of people that would say, man, I want to capture steps. Go back to the steps example, and Kate showed you the picture. We want to capture it for this particular population set, and they would want that to be on the watch. So we would spend time doing that. And when we would release the updated firmware, in some cases it would get released to the actual patient set using the device. But in one example I'm thinking of right now, we had a study that didn't realize the importance of turning off that ability in their settings, and so they got the firmware update also. Well, it changed completely how their steps were processed, and so we learned from that. We were actually able to recover that customer's data because we were capturing the raw data, and I could talk about that too. But when we talk about raw data, we're talking about, hey, what is the sensor telling me that the person is doing from a very unadulterated raw format. When I talk about process data, I'm talking about I've done some operations on that data to turn it into some outcome or measurement. So we were able to go back and get that raw data because we'd captured it and redo it, but it was a lot of work. And Kate, you've had some problems like that, I know.
Yeah, I'm wondering if you're describing a study I worked on when I was a grad student, and we angrily called Actigraph about a firmware update.
I'll take the blame for it all day.
Yeah, no, I mean, I think that it's something that researchers frequently don't think about when they're setting up their study is firmware. It's kind of an abstract thing to some folks, but the reality of it is when there's updates mid-study, this can really impact your data. And same thing with software updates; those potentially could impact your data as well. Another kind of example along these lines is at VivoSense a couple years back, we were doing a study using a patch, an ECG patch sensor to measure heart rate variability in rheumatoid arthritis patients. In mid-study, this technology got discontinued by the manufacturer, and this was a very reputable manufacturer. It wasn't something that, we chose a vendor that was really poor for this. It was just one of these unforeseen things that we did not verify prior to starting the study. We did not ask the question, what's the life expectancy of this product? Is there a chance that it could be discontinued mid-study? And so when that happened, we were left scrambling, trying to find a new product, new technology that we could potentially replace our old one with. And it wasn't a perfect solution. We had to do some performance testing, some validation work for this new device to try to replace the old one. In the end, like I said, it wasn't a perfect solution, but this particular study was a feasibility study, and it was an exploratory endpoint. So we were still able to gather some useful information to inform future studies. And I think the most valuable piece of information was that lesson learned, right? Like, we need to ask that question before we start the study. What kind of support can we expect from this vendor for X amount of years throughout our trial? So
for sure. Yeah, the technology just, it can really mess you up. You have good intentions, but it's the little things. Shamal, tell the story you told the other day about time synchronization. That's a great story, and it's very applicable to what we're talking about.
Yeah, so I mean that's kind of like kind of going back to sort of the validation early development phase of a new digital endpoint. So this is when we were trying to essentially develop a new digital endpoint for a specific condition, and sleep was one of the aspects that we were interested in. And so we started, in most of these cases, when we were running these validation studies, we use multiple devices. Some of the devices are the devices that we're interested in from a endpoint development point of view. Others are more for ground truth, capturing reference data. So in this case, it was PSG, and we also had a video camera, like a infrared video camera that allowed us to kind of see in the dark to look at behaviors that we were interested in capturing. And whenever you use multiple systems, this is sort of a perennial problem. I've since I started working in this space 15 years ago, these problems have not the synchronization between devices has been a constant in my life. And so in this case, what happened was we did a short test, like 10-20 minute test to kind of see if everything can be synchronized across the three systems. Everything looked good, and then we went ahead and pulled the trigger on the validation study. After we get data from a decent amount of participants, we find that there is some weird behavior in the data from the video cameras, which were being used for generating human labels. So somebody was looking at those videos and applying labels. And what was happening was that the video system, the video camera system, had this peculiar behavior of turning off every 20 minutes or so, and for randomly short period of time, and then turning back on, which basically led to an aggregation of a drift, basically. So that the camera was drifting off, and so our labels, which were supposed to be aligned with our raw sensor data, were drifting off, and not in a deterministic manner, right? So this was completely, so basically the choice was for us either basically redo this study or figure out a way to fix this. So we obviously tried to fix this, and we went through this rabbit hole of working with a vendor who was making the cameras, digging down into the firmware of that camera, and it was an adventure. But at the end of that, we basically managed to pull things back to the point where things were usable. They were not perfect, but they were usable. And the cost of redoing it would have been very significant, both from a time perspective as well as resources. And this is something that it's just happens. So it's very, very hard to take into account. So the lessons that we learned from there was, like, if possible, you should always do a full replication of what your study is going to look like. So what we did was we did like a quick 10-minute sort of data collection. If we had done a full night of recording, we would have caught this problem early on and either replaced the camera or figured out a solution, right? And just by not doing that, despite the fact that I've learned over 15 years that this is something I should do, it's still easy to fall into the trap of, okay, everything works, let's go. So yeah, that's my story.
Yeah, and I think a few things there. This idea of doing feasibility or dry run or pilot studies, I know that some people think that those are all dirty words, right? Like that's a waste of time. But the reality is that it's still early days for digital clinical measures, and having, an end-to-end feasibility or pilot study cannot be undervalued, in my opinion.
Yeah, just to add, Kate, I fully agree. And I think when we are deploying technologies into clinical trials, I would argue that you should test the whole workflow, not just a piece of it, but actually, all the way from the first point A where you start to the point, the end point where you're going to get the data back from, into the hands of your statisticians or analysts, whoever. I think you have to test the whole workflow.
I think it's super important for people to just get permission to don't be afraid to ask the dumb questions. Sometimes people are like, well, of course these sensors probably have done this thing that seems entirely obvious to do, but time is actually really, really complicated for a lot of things to work on, and getting a couple of those things right, whether it's the time example that Shamal talked about, or even just testing. Like we were talking to one group and they would take both sensors and just drop them at the same time and then look at them and see if they registered or do certain things. This will just I think, pay off in the end as you're going through that sort of stuff. I think there were a couple examples, not only in the sensors themselves and checking the kind of the specs of the sensors, but Jeremy, you had some examples of how you're setting up some of the resolution and how you might want to think about it. Because then when you're pulling that data off, what might that look like?
Yeah. There's actually a great question in the Q&A. I just was looking while you guys were chatting. It's talking about scratch. Is the technology good for assessing scratching And then the next part is it good for assessing sleep? Is a methodology established for assuring adherence? So it's in the root of this question. I think people, I don't mean to generalize, but we see it all the time that the technology selection happens first. And I know I want to use this device because I'm excited about it and I know the patients are going to love it. And I think there's something to be said about patient engagement. It's very important. But this is a great example, Andy, that you've mentioned and that this question alludes to, and that is fit for purpose, right? We hear that sort of term thrown around a lot, and we just, kind of put it in the back of our mind as, yeah, I need to use somebody who's fit for purpose. I'll think about that and ask Jeremy when I call them if they're fit for purpose, and I'll check that box. A good example of what that means is, let's just talk about scratch, or even let's talk about essential tremor. We get this question all the time. I want to have my population set wear an actigraphy device. I want to measure their general activity and also want to look at essential tremor. And I'm thinking, man, that's great. We don't do that. and what do I mean by that? We could do that, but let me explain what I mean. So our device is set up to measure an 8G sort of swath of motion. So 8 units of gravity I can measure. By the way, a Blue Angel pilot around here in Pensacola pulls about 7.5 G. So that's sort of the range we're talking about. That's actually a good range for general activity. It allows me to see all sorts of movement in the day. It allows me to see if you had any type of heavy hitting intensity activity, if you did a workout. But what part of the issue is that if I want to look at essential tremor, I need to get just higher resolution around a very small range. So I would want to switch the device into a mode where it's not measuring 8 Gs, it's measuring 2 Gs, right? That way, my resolution on the measurement that I have there is going to be much higher and tighter, and I can actually see the little fluctuations in that tremor over time as the therapy is applied. I won't be able to see that at an 8G resolution. That's what we're talking about when we say fit for purpose. And that's why it's not just about the wearable device, it's about the setting and the preparation that happens in the back end. And in our case, we have a tool that before your study starts, we walk through and we say, hey, this is how it's going to function. If the patient presses the button, we're going to jump into this mode and allow them to test that, or maybe we're going to do that twice a day automatically. But those things have to happen without the patient's interaction, because otherwise it's just not going to happen. And by the way, if you aren't able to do that, you're not going to capture what you expect to capture. And so we see that a lot, and it's important to understand that fit for purpose is not just about picking a device that chooses that. It's about picking the device plus the actual settings of that device, plus the platform, plus training. Those are all part of the big picture. So it's just a good example that addresses the question there too.
Yeah. So if you think about this, none of these sensors know if you're sleeping, right? They don't know if you've taken a step. What they're doing is they're taking maybe your accelerometer, gyroscope, height, weight, and age, and then predicting whether or not those things combined into that. And as you're, like all of these are just predictions, right? That we then classify into something. So sometimes as we make these predictions, maybe the setup of the sensor isn't the right setup to get that prediction in the right way. Sometimes actually the hardware itself just kind of messes up. And I think, Kate, you had a couple examples of signal artifacts where you were like looking at it, it was a little bit funky, and then you had to do the murder mystery sleuthing into what's going on there.
That is true. I'll share my screen again and show a picture of that. Yeah, so let's get this out of here. So I'm assuming you can see that.
Yep, that's great. Yeah,
so this is actually an example that was shared with me from a colleague, Rick Troiano, who's at the National Cancer Institute. And these data come from the NHANES surveillance studies, so the National Health and Nutrition Examination Studies. So this is a huge surveillance study done in the US where they deployed Actigraph sensors on, upwards of 16,000 youth and adults. And when they had all these data at the end, this was a tremendous amount of data that they had. They were doing their QC checks on it, and they found a couple of examples of where the signal is just not believable. So there's some sort of artifact that's causing the signal to essentially misbehave. It's this idea that the signal is registering something that it shouldn't be registering. So in this example, we have, the sensor is stuck at a continuous max. So it's just reading the maximum Gs it can for, a minute to hours at a time. And then there's another example of where it's kind of doing these interval fluctuation or jumps where it's just, repeating between this consistent pattern, which again, isn't believable for human movement. And then the other example over here is these instantaneous spikes where the change in acceleration from sample to sample is, again, just not believable for human movement. And so these were unexpected artifacts, but yet the team knew that, this is real world data. We can expect there's going to be some sort of artifact in the signal. So we need to do some QC on this. And so there's a lot of different, signal processing techniques that you can use to clean artifact out of the signal. There's other techniques that you can use depending on what type of sensor you're using and what your context of use is, what the measurement setting is, those sorts of things. But the point is that when you use a sensor that is collecting data at high resolutions in the real world, you should expect that there's going to be some level of artifact, whether that's due to a technology malfunction or whether that's due to something internal to the patient. So for example, if you're measuring ECG or respiratory signals, when that patient is moving and or coughing, speaking, crying, those sorts of things, that could cause artifact in your ECG or a respiratory signal. So expecting that in the real world, these things will happen. And I'll just note that for this example with NHANES, they had, almost 17,000 accelerometer files, and it was a very small amount of the data that this sort of artifact impacted. It was less than 1%, something like 0.2% of the data. But, depending on what your use case is for the data, it could really impact your outcomes. So, planning ahead to have some sort of QC process of any data collected in the real world is a really good idea.
Just to be real with everybody here, this is our data. This is Actigraph data, and we were chosen for the NHANES, and so this is a real problem. And this was back in 2013 when this technology was deployed. And so literally, just to kind of show you what we found out is happening there. This is the inside of an accelerometer, like a digital accelerometer. A lot of folks don't realize what's going on inside, but there are literally masses and springs inside this MEMS. It's a microelectrical machine essentially, and these plates move around, and you can kind of see a close up here, some of the masses and the springs and the capacitor plates. So for this particular accelerometer, I'm not going to throw the vendor of that device on it. Like it literally had a bug in which it would stick at a maximum value, and it would dislodge itself over time. But the idea that you could catch that even in verification of the device, is just very random and very sporadic. It's just going to be there. So we've seen that. I mean, obviously this is fielded data. Fortunately, it affected a small percentage, but the idea of rescuing it, there are some things you can do to rescue it, but you just need to plan for that. That's just going to happen. So just to share and be transparent with everybody, that's on us. We did that. So.
But I mean, to be fair, right? Like that's to avoid artifact due to a technology malfunction is virtually impossible. Like it needs to be expected that it's going to happen, but here's how we're going to try to avoid it, and here's how we're going to handle it on the back end if it does.
Absolutely, just planning ahead, right?
I mean, there can also be, non-technical issues. So, for example, one of the studies we were running, we had devices one device on the chest and the other device on the thigh, sort of the upper thigh. And we were using those devices, the two devices, essentially data from those two devices was being used together to decide what types of activities you were engaged in and some other metrics. And this is entirely plausible, but we didn't anticipate this. Was one of the participants switched the two devices. So the device on the thigh was now on the chest, and the device on the chest was now on the thigh. And so all our predictions were like flipped, right? So for example, if we were detecting sitting, and now we were detecting that you were standing or lying down, depending on what orientation the device was in. And those types of things just are sometimes hard to control for. But if there might be ways to mitigate it, both from an analysis point of view. So if you see a lot of sort of in this example, if we saw that you were the participant was lying down for a significant portion of the day and up at night, that might indicate something, sort of that there must be something that needs to be looked into. But this one is a tricky one to kind of figure out.
Shama, one of the Q&A people just wrote, the participant really loves doing handstands. That was really what was happening there.
Yeah. In that example that Kate showed, like you could, what Jeremy mentioned about the fighter pilot, right? Like that flat line could be somebody skydiving, but the chance of that happening is very, very, very low, right?
Yeah. And these things happen all the time too. Like we're talking about the sensors, then we're picking the wrong thing. There's so many things on the usability testing. One of my other favorite ones was somebody was testing out a way of doing a 6-minute walk. And then they were supposed to take the smartphone and put it in the pocket. But many women, traditional women clothing doesn't have pockets. So the person didn't have a pocket and just put it on the table and then walked around for a little bit. So clearly that didn't register anything for the event. And then there is another one where they're looking at tremor, and it was performing very, very well in the clinic, which they're really excited about, especially for like the sensor could actually pick up things that maybe like more doctors might have been more distracted at seeing, or the clinicians might have been more distracted at seeing. They're like, great, let's send people home. And then all of a sudden they were getting like 10 times more events and trying to figure out what was going on. And it was in the Netherlands, and everyone bikes on cobblestone. And then that ended up having like a whole nother set of things to consider. Before we jump into a few more of the Q&A, any other last ones, Kate and Shamal and Jeremy, that are just like favorite ones?
Oh gosh.
Just wanted to kind of play off of what you mentioned, Andy. So when you put device, I think that the one thing that I've learned over the years is that when we take technology and deploy it at home, the problem, I think that the natural tendency is to measure continuously. We want to get a lot of data continuously, right? And what I've learned is that what we actually want is to capture measurements at the right time, under the right conditions, right? So your example of cobblestones is great, right? So like if you had, sort of anticipated that we want to capture, data maybe in the home setting, and using geofencing or some other technology, that's where the tremor information is most meaningful. Then you could get, you could deal with some of these issues. So in general, I think using context to drive the measurements, the digital measurements in the free living setting is the most advisable thing to do.
Yeah. And Andy, I have one more just horror story that I'll tell that actually changed how we did business at Actigraph. We had our watches, they're manufactured in the US, and they come to us and we do our quality checks. But when they're manufactured, we do this process to calibrate the device. And we actually put it in 14 different planes to ensure that no matter how we're moving it, if there's if I put it at an angle like this, the total sum of acceleration should be 1G, right? I mean, we live on Earth, so it's 1G. So we actually had a series of devices that during that process of calibration, the offset, said that it was measuring 0.92 Gs, and so we added 0.08 to it. And then we told the watch itself, we said, hey, anytime you see a measurement, add 0.08 to it, and that'll adjust in that realm. Well, it turns out that was an error. It actually calibrated incorrectly. And so we had something like 500 watches that got fielded, and they were miscalibrated. And so all this preaching about raw data that I do was for naught, because the raw data itself was flawed, right? Because we calibrated incorrectly. So we actually changed our process. Now what we do is that when the device gets calibrated, we don't store the calibration offsets on the watch itself. We actually store them in a database. And then when data comes into our system, we then calibrate it as it comes in real time. And that way, if we find that problem in the past or in the future, we can go back and say, hey, these 300 devices, they were calibrated wrong. Let's go back and reset that offset and change that value. So actually, when I talk about raw data now, I'm literally talking about pre-calibrated raw data. That was a big game changer for us, because we're thinking, man, that'll never happen, right? But we ended up with some data issues because of that. But we called the customer and we said we got a problem, and I'm sorry, and that was not recoverable, by the way. But fortunately, the adjustments that we were looking at there were minuscule, but just a scary time for us, honestly. Anyway.
So I do want to jump in with one last thought, because we really haven't, I know we're running low on time and this is kind of a can of worms, but we haven't talked much about failures, I think at an operations level, like a ClinOps level. And I'll skip right to the lesson learned in that. I think that, again, I said this earlier, this is still early days for using these sorts of tools in clinical trials. And so the idea that the operations staff may not understand how certain decisions impact the data is real. Like, this happens all the time. And so the number one thing is obviously having, good systems and training in place up front, but also it's not a set it and forget it to have the right level of engagement with your operations team throughout the course of a study to verify that the right procedural steps are being taken throughout, I think is of utmost important to avoid data going awry. So.
Totally. Okay, so what we're going to do next is, first of all, I'm super impressed with this group. We have so many people in the chat, so many people in the Q&A, and I'm like a nerd for all these questions, so I would want to like stay around for hours to do this. The way that we can do this is we are going to finish a minute or two early, so you can all do a break before your next Zoom calls that you're going to do. Each of the four of us are going to put our preferred contacts or some type of contact in the inbox of how we may or contacting our companies, if that's the best way to do it. And then we're going to pick a couple of the questions. There's so many more that we want to do. Either reach out to us directly, or maybe we have more of these. Maybe when COVID's done, we actually rent this place and we can we all can hang out together, attendees and panelists, to talk through this stuff. And I do want to remind everybody that this is one of the most important things for us to work on together, right? If there's a place where you can actually be pre-competitive, like nobody's trying to compete really, I hope, on understanding like how human bodies are, operating. Like, I think it would be a very bad future for us to have meaningfully different ways of measuring steps that weren't like clinically relevant, but just because different companies were thinking about it differently. And so it's super important to me that we have an intentional and thoughtful way of doing this. And I'm sure regulators really don't want to have like a massive proliferation of endpoints either that doesn't have a clinical reason to do so. So I know I might be an optimist here, but like, let's work together. It's early days. We'd love that. It's fun for me to work with the three people here, and we'd love to work with you too. The Digital Medicine Society, which is a nonprofit, is a place that you can join. It's a 501c3, it's dimesociety.org. That's one other like home that you can have to start having more of these conversations. And so I guess for three panelists, type in how you think a good way of staying in touch, either company or personal. And then I'll pick a couple questions for people to go through. So one of the questions, well, I think one question that we haven't spoken to, we've done a lot of technical horror stories, but these sensors are a very significant surveillance opportunity, right? Like, people are trading off possibly some civil liberties around being able to collect this sort of data. How do you think about those trade-offs, and how do you think about data rights and privacy? This is obviously a much bigger question than just this. These are entire teams that work about this, but what's like a first set of thinking about that?
Well, yeah, I think we give away some of those rights anytime we participate in a clinical study, whether it's with a wearable or not. But certainly when we're moving data to the cloud, it's a difficult question, Andy. I mean, I think each vendor handles that in their own way. We do our best to ensure that. I'm not trying not to market here, but I think there are certain checkpoints along the way that have to happen. When we're audited, we have to prove that these systems are isolated with no access by anyone other than the administrators and the study leaders. We have to show that we're under BAAs with our partners. We have to show that we're doing the right HIPAA controls and checklists along the way. So processes and procedures that are in place, but admittedly, like there's no golden way of fixing everything with regards to that. So.
There's probably everybody has a thought for this, but I'll pick another question. And these are just teasers for future conversations that we can have. Okay, so either Kate or Shamil, do you have a view on potential biases from displaying the data? So should a participant or study participant be able to see that information? Does that help engagement? Does that change the study? How do you think about that sort of design question?
Shamil, I'll tackle this one first and you can chime in. I mean, I think like all the answer to this question is the same for everyone. It depends, right? Like, what's the study? What are you doing? Of course, when you put a new device on somebody, they may be like, if they're getting feedback from the device or not, even if they're not getting feedback from the device, they could be inclined to change their behavior because now they're being monitored or now they're being monitored and getting feedback. So that is something that needs to be carefully considered within the context of use. What is the question? Of course, if you're trying to do an intervention and change behavior, then that's a whole different can of worms and you're trying to use the tool to actually implement the intervention, then that's something else. The other thing around that is, you know, if you do suspect that your patient population will change their behavior because they have something on their wrist now, and you're trying to get a good baseline measurement of what their habitual activity is, for example, you may decide to monitor them for longer at baseline. So maybe 2 weeks instead of 1 week, a month instead of 2 weeks, because the idea that they're going to change their behavior and maintain that behavior change over that baseline period, it reduces as time goes on, right? Like behavior change is difficult. If all we had to do is put a device on somebody and they would all of a sudden become more active, then that would be fantastic, but that doesn't happen all the time. So I think just being really thoughtful about what your study is, what question you're trying to answer and who your population is, that's the approach to take.
Yeah, I think that's great. I don't have anything to add to that. That's, yeah.
Perfect. Okay, so I think we're wrapping up for, I guess, let's just do maybe a quick round robin where Jeremy, Shamil, Kate, like last thoughts for people or wisdom that they might have as they embark to use more of these tools and create more connections for, you know, how somebody's living in a natural setting at home.
Yeah, I'll start. Don't reinvent the wheel. There's plenty of resources out there and plenty of help. I mean, you got a panel here where we just want to help folks, and so check out some of the resources that are out there. Andy's posted some in the chat here, but you know, ask questions and start early. Don't you know, hey, I'm going to add this into my study, you know, a month before I start. Start planning early.
Yeah, my 2 cents would be, you know, create a process, develop a process for how to, you know, do this, you know, identify the technologies, identify the right measures, and then the operational side. I think essentially like what has been outlined in the playbook, you know, use that and develop your own processes to have, you know, from a consistency point of view, but also making sure that you're not going to just do things because you've done things previously, right? You have to kind of take a new problem and try to really understand it and go through that process, even if it might seem obvious that you might want to use what you've done before, right? That's.
Yeah, and I'll just reiterate that the playbook and DIME in general are fantastic resources for people, whether they're just starting to get into thinking about using these sorts of tools or whether they've used them for a long time. So definitely those are very, very valuable resources. And my take home message, I think, is this isn't a one size fits all approach. Like we've said throughout this, right? Like every specific scenario deserves, you know, well thought out procedures, well thought out, you know, risk mitigation procedures, all sorts of things. So I think, as Shamil said, yeah, you need to think through your question and find the right solution. You can't just have your solution in mind and apply that to all questions across the board.
Amazing. All right, thanks for joining for our fireside chat. We hope to have many more of them. We think there's a very exciting future here for healthcare, and we hope to join you in your own adventure on this. So thank you.
Thanks.
Bye.
Thank you, everyone.